Good morning, children. Welcome to biology class. Today we are going to start chapter 6, genes and chromosomes. Children, Gregor John Mendel, he assumed that characters, they are determined by certain factors of inheritance, which are stable, particulate, and they get separate independently at the time of gamete formation. But he was unaware of these factors where they are located in the cell, and he also not aware of the details of cell division. So he could not identify physical counterparts of these factors. The role of nucleus in reproduction and existence of chromosomes, these were also unknown to Mendel. Development of new and improved techniques subsequently helped in the discovery of chromosomes and the process of cell division. So let's start with the chromosomal theory of inheritance. So after the discovery of Mendel's law in 1900, Walters and Sutton and Theodor independently observed behavior of chromosomes and Mendelian characters. They noticed several similarities between the chromosomes and the Mendel factors. Like chromosomes, they occur in pairs, like the alleles of Mendelian factor. The homologous chromosomes, they get separate during meiosis, like the pair of similar or dissimilar alleles of Mendelian factor. Different chromosomes orient and separate independently during meiosis, like in Mendelian factors, and the paired condition of both chromosomes and Mendelian factors, they are restored during fertilization. And Mendelian factors and the chromosome, they maintain their individuality from generation to generation. Now these similarities which Sutton and Boveri observed leads to the chromosomal theory of inheritance. Now this theory states that Mendelian factors, they are located on chromosomes, and it is the chromosome that separates and assorts independently during meiotic cell division. So here the simplified version of chromosomal theory presented here. It shows the movement of alleles of two genes, seed shape and seed color, which are located on two different chromosomes during a dihybrid cross of round yellow and wrinkled green pea seed varieties. Now the two duplicated and paired homologous chromosomes, they assembled at the metaphase stage of meiosis 1, and they can separate in two possible ways. So these are round yellow seed, and these are wrinkled green seeds. So through meiosis 1, they separate in two possible ways, round yellow and wrinkled green. Now this leads to the independent assortment of the chromosomes, and the genes, they carry in four possible ways. So after fertilization, so they carry the seeds, all round yellow seeds. Okay. So in F1 generation, now after meiosis 2, so after meiosis 2, 4 types of gametes are formed, each with half number of chromosomes and genes. Now the 2 parental types and the 2 new recombinant are formed in equal ratios. So here this metaphor is 2. So the F2 progeny, they exhibit phenotypic ratio of 9:3:3:1. See here, yellow, round and yellow, wrinkled and green, then yellow and wrinkled, then green and round. Okay. So in F2 generation, after fertilization, the ratio is 9:3:3:1. So here these are round and yellow, these are wrinkled and green, again wrinkled and green, and round and green. Okay. Now come to linkage. Now linkage, so Sutin, after commenting on the chromosomal theory of inheritance, indicate that since the number of genes in an organism, it is much larger than the number of pairs of chromosomes, and each pair should contain several genes. It means that during meiosis, chromosomes, they move as a unit, and all the genes of a chromosomes, they go together and they do not assort independently as expected according to the Mendel's law of independent assortment. Only those genes which are located on different chromosomes, they pair assort independently. This also implies that the genes which are situated on the same chromosomes, they should be transferred en bloc from one generation to another. So this group of genes which behave as a unit is said to be linked, and the phenomena it is known as linkage. So all the genes on a pair of homologous chromosomes, they collectively form a group known as linkage group. This indicates that the genes which are linked together, they are situated on the same number of chromosomes. For example, Drosophila melanogaster, it has 4 linkage groups which correspond to n, which are equal to 4. Now the genes which constitute linkage group, they are inherited together and they are not independent. Thus linkage, it may be defined as the tendency of 2 or more genes of the same chromosomes to remain together during the process of inheritance. Types of linkage. There are 2 types of linkage, complete and incomplete. So complete linkage, it exhibited when the genes for a particular character, they are present very close to one another, And this is because of non-break in the genes combination which are situated on a chromosome. It is not very common. An example is male Drosophila for grey body color and long wings. Now Morgan, he demonstrated through his experiment on Drosophila that a cross between the male with grey body and long wings and the female black body with vestigial wing. Two recessive characters, black body and vestigial wings. In F1 hybrids, which are like the male parent, if a male from F1 generation is black back-crossed with a double recessive black vestigial female, the four kinds of offsprings in equal numbers as a result of independent assortment are produced. So only two types of offspring which resemble the two grandparents. Now this suggests that the genes for grey body and long wings are linked together and they show complete linkage. Now incomplete linkage, it occurs when the genes are of different characters. They are separated at the time of gamete formation due to breaking and exchange of chromosome pieces during meiosis. In sweet pea, blue flower color and long pollen character, they exhibit incomplete linkage. When a sweet pea variety with blue flowers and long pollen is crossed with another variety having red flowers and brown pollen, individuals produced blue flowers and long pollen in F1, and these F1 individuals when again crossed with plants having red flowers and brown pollen, we find the test Ross frequency in this table. So, whatever ratio is expected in independent assortment, but instead of 7:1:7 ratio was actually obtained, indicating that there were a tendency in dominant alleles to remain together. So factors affecting linkage. So the linkage, it is affected by several factors like distance. Strength of linkage is inversely proportional to the distance between the linked genes. Age of an organism. X-rays. It reduces the strength of linkage. Temperature, increase in temperature decreases the strength of linkage as the chances of recombinant increases. Now the significance of linkage. The phenomena, it has great significance in reducing the possibility of variability in gametes unless crossing over separated the linked genes. So crossing over we'll learn in our next class. Till then, thank you and have a nice day.